Okay, thank you. This is, I just showed this slide. It's not related to the lecture because I mentioned it after Luke talk. And this is the exosome, something that relates to another mode of communication in the body that cells, normal cells, cells in the brain, and also cancer cells send small nanoparticles that have DNA and RNA and microRNA, and they can go to other parts of the body and induce epigenetic and genetic changes. So that's very important for understanding. And I think, I don't know, but I think that the properties of water in the body has a very strong effect on the way that they work, so it affects the communication. Just to show you one picture of exosomes, this is not my picture, it's a picture from someone in the Technion. I think this is a striking picture. What you see here is a breast cancer cell, and you see this little sphere. It's like a science fiction movie. They are ready to be launched, and what you see here are nanotubes that are connecting these cells with other cancer cells. So they do it in a collective way, so they can secrete a large mass of these nanoparticles or exosomes and affect other parts of the body. Okay, so this was just, but Luke is not here, so he didn't see the picture, but I can send it to you. Okay, so the topic today is about, look, you are here, so I'll show you the picture. Sorry, where is it? That's the cancer cell I just mentioned with the exosomes that I promised you. And these are nanotubes that connect between other cells, and they do it in a coordinated way. Okay, what I want to talk today, everybody talks here about rethinking water. I take it from a different perspective, and this is something very simple, but maybe it has some important consequences. We did the study, and we were surprised to discover that natural water contains nanoparticles at a density that is not high but significant, about 10 to the 12 particles per milliliter, which means that the distance between them is of the order of half or 1 micron. And since this is in all natural water, they of course can contain different particles, so they have some different properties. And since life started in water in bacteria, and from bacteria we are, I think that we adopted to respond to information which can be stored in water according to these nanoparticles that induce order in water. So this explains maybe part of the puzzle. Why is it that we do different things like adding particles, adding electromagnetic radiation, and the biological systems know, in quotation, how to respond to it? I think it's something that evolved since the origin of life, because this is what happened in the origin of life. We had both these nanoparticles and very high electromagnetic activity. So the take-home message and the outline is, I first show you that they really exist. Even this simple thing, we talked about being speculative. Usually I consider to be out of the box. Here in this meeting I feel very much inside the box, because even this thing, just the existence of these nanoparticles, we had a hard time to publish. That's a fact of life, how much we talk about the objection of scientific community for things which are new. The other thing is that the existence of these nanoparticles, I show you very short, very briefly, because some of it I showed before, and it's the last lecture, and we don't have time. I want also to communicate some ideas. They have singular effects. And the word singular means, and this is essential for, I believe, all that we do here, is the idea, and this is mathematical idea, but has to do also with physics of open systems out of equilibrium, that when you have small, minute effect, it can lead to a large response. For example, the first time that I encountered to it is in the formation of snowflake. When I was a postdoc, we were puzzled by the question of Kepler from 1610, how is it in this homogeneous atmosphere, you have very nice structured snowflakes. And what we figure out mathematically and then experimentally is that the very minute effects on the interface between the ice and the atmosphere are amplified by the fact that the system is driven out of equilibrium and they end up in building this beautiful, highly symmetric structure. And I believe that this is something essential that should be part, if we one day we turn what we're doing here into a new discipline, this will be one of the elements, very important elements of the theory. So I show you some singular effects, for example, on pH, participation, electromotility, and mainly loss of bacteria. I wrote here theory. We don't have a theory. What I write here, a theory, it's not a theory, I forgot to put the quotation. What we have are thoughts or reflections of what might be. We have ideas, but we don't have a theory. It's like quantum mechanics before Schrödinger equation. So we had many different observations, many different pictures, but we didn't have a theory. We don't have a theory, so when I say theory, it's a teaser. We don't have a theory. I wish we would have a theory, but we don't have a theory right now. And the elements of these observations, and each one gives different names to different phenomena, I have to do, and we agree here, that there is effect of a network, formation of network, of nanoparticles, nanobubbles. Some people call it other names, structures. It's hierarchical. We have a cascade of length scales, so it's not one scale. They act as a singular perturbation, and what I try to use the term is systemic view of water. That water behave in a collective way, and water is far from equilibrium. Otherwise, I feel not that I know how to explain it, but we cannot explain it otherwise, because it contradicts what we know from physics. And then I mentioned some reflections on the body. We heard some wonderful lectures about the body. Some of them I agree, some of them I don't agree with some of the claims, but nevertheless, clearly there is effect on the body. Okay, towards systemic view of water, because it's the last lecture, some people will live in the middle. What I mean by this is the artist concept of the network. I want to take home message to be this before I start the lecture. From entropy to information. I think we mix all the time, not only this field, we mix also in physics, in other sciences, the notion of entropy and information. Since Shannon that tried to define information, but it's not information that we need. He mentioned information in the sense of capacity to carry information as minus the entropy. And what, for example, in Luke lecture and other lecture, when we talk about information, we talk about real information, that you have specific information, not the capacity to have information. So there are two different things. Today in physics, we think that entropy is a real quantity, but some of us it means like 3 people or so, or more, but this order of handful, talk about the fact that information can be ontology of reality of systems which are out of equilibrium. Then we talked about information storage and transfer, but first of all, before we talk about this, we have to talk about information. If we have time, I'll reflect on it, because to store information, you need energy to change the state, and I think there is one of the big puzzles of what we do is what is the source of energy that if you have a minute electromagnetic wave or add minute amount of particles as a medium to transfer the changes, well, is the information that can imprint it, because each bit of information needs an energy which is at least 10 times k Boltzmann T times 1, 2. Okay, one of the ideas that and again, it's idea, it's not a theory, that comes to mind is that there's something like a prion effect. So you have some structures in water, there can be structure in different topology, and then when you trigger something, the electromagnetic wave, another thing, it acts as a catalysator that causes these things to assemble in a way that eventually will be irreversible. Otherwise, it's very hard to understand how it can be that we have such minute signals and they imprint something irreversible. Okay, so that's just to share with you some of the things. And that we discussed with Luke many times. He asked me questions. Sometimes I understand them, sometimes not. In return, I give him answers. Sometimes he understands, sometimes not. Okay, water is the fabric of life. The idea is very simple. Life were generated in water, so they resonate with water. Resonate not in the terms of resonance in physics. This is just to show you one example, which again is fascinating thing. It's a spore of bacteria. It's the closest thing to be between alive and dead, okay, because they can endure forever. And one of the puzzles is, how can it be that it's so stable? People took it out of space, exposed them on a shunt without safe condition, brought them back and germinated them. So they are very, very due to heat, many other treatments. Yet you put them on a soft agar, and somehow the information is transferred, and the spores know to germinate. It's not a chemical transfer. Personally, this puzzle is the thing that triggered me to think about transfer of information without transfer of chemicals. Okay? And indeed, our recent studies that the structure of water inside the spores is very special. Okay, so structure of water in normal cells and cancer cells is different. If I have time, I'll reflect on it. Chemical reactions in natural water are different. Chemical reaction in our body, of course, is different. And the activity of molecular engine. If we have all the water, then because the entropy is different, all the activity of the molecular machinery. Just to give you an idea, people talk about the membrane of cells, membrane of bacteria. 85% of the genes of the bacteria are dedicated to control the membrane, because that's the essence of the life, is the exchange between the inside of the cell and the outside of the cell. Okay. For bacteria that are out, we heard about it in the talk. We heard about the guts. We have here 10 times more bacteria than we have cells in our body. About 40,000 different species, and the number is growing all the time. They affect many things. Every day there is more things that have been discovered that they are affected by the gut bacteria. They have a very special communication with our body and our brain. You have about 100 million neurons in the guts, much more than what you need for the mechanical world, and they have a very strong connection with the brain. The number of neurons in the gut is about the same as the number of neurons in our limbic system, just to give you an idea. Okay, so this is one thing, and we heard about it in the lecture. Mitochondria. Mitochondria, we heard also, it's a colony of bacteria in each cell of our body. Many or most people are aware that they are the power plants of the cells, and that's why it's so important. Power and energy is important. Unlike usual thermodynamic machines, what we have in cells is that you produce coins of energy, and the coins of energy go to specific places to do the activity. So you can fight, or you are not subject to some of the limits of the second law of thermodynamics, because it goes according to information to be inserted in specific places. People don't know that the mitochondria are in control of regulation of all the sex hormone and our brain, activity in the brain, and energy management and cellular differentiation, which is essential for understanding the differences between normal cells and cancer cells. I don't know if I get into it, but I think we have some, in this case, some nice understanding that it's not only ideas, but concrete things that we can show. Okay, eventually, in the reflections, we heard a lot about healing, different way. I hope I'll have time to show you. I was puzzled by this fact that I tried it myself, that people will do healing, Reiki for example, and I could sense the effect. It puzzled me as a physicist. The usual solution that it's all placebo, but it seemed to be that it's real, and I tried to study. This is initially the idea, and I show you some results with Miana that did afterwards, that you put your hand near a test tube and you can detect the effect. This does tell us how the fact that it does induce changes in water can help healing. That's a whole distance from there, but in principle, it can. Okay, let's go fast. So this is the natural particle, so this is the size, and they mainly calcium carbonate. We can see it because we use a special tank in the Technion that is very expensive. I forgot to mention one thing, that part of my exposure to the field was thanks to Grander company, without me knowing who is Grander and who is the company, because we had a friend who is a distributor in Israel, and she showed me some strange result that a cat goes to water that were treated, and I think to me that this is crazy cat, and in physics we like crazy cats in Schrödinger, but and we just like cats because my wife is a witch, my mother is a witch, my wife is not a witch, my mother is a witch, and my mother used to try mushrooms, if they are good or not by going to Google, but by exposing to the cat. I waited until she would test it, and then I would test it. But anyway, so this is also tap water. I have nanoparticle. This is tap water, actually quite clean. I was very surprised. We took tap water in Israel. Forget the fluorine and the taste, but in terms of nanoparticles, they were very clean. Beautiful. This is a close-up. It's a very special substrate that you can have some of the particle attached like this, so you can have the beam going through, so you can have the chemical content. You see here, you see also that they have some shell around them. They have some internal structure, which is fascinating. This is just to show you from Italy, San Benedetto, and you can see by the peaks that it's mainly calcium carbonate with some additional things. Okay, this is two types of water from the Alps. This is from Grander. One of the things which are fascinating, I was there in the Alps at the same location, and this structure here, which is very puzzling because we saw stones on the microscopic level that look the same. But this brings me to the point that things that look similar are not necessarily the same. We are sometimes subject to a reminiscence complex. That if we look at some pattern and we say this explains this, not necessarily. Okay, this to show you fast freezing that they are really distributed around. And this is a surprise by itself, because you'd expect that if you freeze them, they will collapse. So there is some exchange interaction between them. Okay, this is to show you another thing which is relevant to the US, because everybody in the US drinks purified water, and purified water do not have the nanoparticle. And they have very different properties. I'll show you in a minute. And water with salts, you see, because some of the referees say that this is what we see, just aggregation, normal aggregation. This is normal aggregation of salts. It does form this particle. And this is seawater. After looking at this picture, I think twice before going to the sea in Israel. How did that turn here? So now we go to the singular effect. Okay? Just how are you doing your time?
We have about 20 minutes.
Okay. So just to show stabilization of pH. Someone gave a lecture about ionized water, and he said that tap water have about 5.5 pH, and it puzzles me because the water that we tested have about 7.5 pH. What you see here, you dilute the water, so the content of inorganic carbon changes, okay? And nevertheless, the pH stays the same. Now, if you take normal water and you add, for example, some salts, carbonate salts, to increase to the same level of inorganic carbon, the pH goes very high. And this is just natural water. It's not any special treatment. That's what happens. So it seems that natural water with nanoparticles work as a buffer for pH that might be very relevant for life. And we know that bacteria in our body, 7.5, around 7.5 is the working point of life, of pH. You can see electrophoretic motility, zeta potential, and you see that natural water and still water have a very different thing that shows you that the chemical reactions will be very different. This is the old work that reported before. This is with Yael Katzir that is here, and this is electrochemical deposition that you have electrode around, electrode at the center. This is the deposition. This is kinetic effect. If you have control, you make zinc sulfate with normal diluted water, and you have very high level of zinc sulfate. So the salts in the water will not have an effect. And then you do it with nanoparticles. This is what you see. So there is a dramatic effect. That's the thing about such a difference. If you look inside, there is a connection between the macro level and the micro level, and you see here you have some organization, which we think is a reflection of the organization of the water. It's a reflection. We don't know how to explain exactly how it is formed, but it definitely shows that there is something that maintains long-range correlation relative to this case. Okay. The thing that's really close to me are the bacteria. As a physicist, I started 25 years ago to work on bacteria colony. The motivation was not water, the motivation was to check if bacteria have the origin of intelligence and cognitive functions. But now there are too many things. I developed 2 strains of bacteria, a new way to grow them. 2 strains, this is Bacillus dendritiformis. He has this very beautiful color, special color shape. We have another one that is Bacillus vortex, we call it, that it has these dots, and these dots are a group of bacteria that turn around. The reason that it's so important to work with these are wild strain, so they are not strain that domesticated, that lost many of the interesting genes over the generation that has been grown in the lab. And the fact that they have such nice pattern enables us to use them as a probe of many of delicate things. However, and that's crucial, because they are so sensitive, it is extremely hard to have reproducible experiments. If you so the way that we do the experiments, we grow the bacteria in different petri dish, or every time 10. Even we number all the plates, because there are so many differences between the plate that is here and the plate that is here. And I develop all kind of method to keep the condition the same, and I show you the level of reproducibility, because it's essential in order to draw a conclusion. This is just to share with you the beauty of it. Some of you didn't see it before and interested in the beauty, so just try to enjoy it, although there is the pressure of going to the airport. This is artist concept of this bacteria to grow pattern that artistic. You don't try to make it reproducible. You actually want different ones each time. And you can just enjoy it to give you an idea of the complexity. Okay. Now, you're convinced that they are beautiful and smart and they can be probed. So this is what we mean. This is distilled water. When I say distilled water, what I mean is that even if you take the bacteria, make the gross egg beads, the broths that they like to grow overnight, then you take the bacteria and inoculate on a plate. They still keep the memory of where they conditioned before, and you see this is the level of reproducibility between 2 plates, but we have more. It's essential. And then if you do the same thing, you take natural water and you make the broth that you got bacteria, and this broth has a lot of salts. So the salts in the natural water are meaningless. It's the nanoparticle, and you take and look at the difference in quantities. This is to show you that it has an effect. This is another. This is the other strain. You see the difference in the pattern. Now the other thing that we did, and this has relevance to the bacteria and so on. Many people talk about water, different energy water, functional water, and so on. And we wanted to see if there is something in it. So what we did is to make a substrate with 50% distilled water, 50% different type of natural water, mineral water, and we grew the bacteria, and you can judge for yourself. We have a repetition. I don't have time to show the repetition, but this is, for example, one example of one type of water. Now, if you look at it, this is a control, one type, another type, another type. And these are two types of water from Austria. For some reason, the water from Austria is more tyrannical. Okay, now this is just to show you the level of salts that you have in the natural water, and this is the amount of salt that we add to the water when we make the substance. Okay, so we now go to the theory, not a theory that is not a theory. Okay, so the idea is the physics of self-organization that I played with it in other contexts, and it is now quite accepted, not for the water, but as a new type of physics effect. And here is what we proposed time ago, the formation of network of nanoparticles. When I say nanoparticles, I mentioned before, we have to work on the definitions, okay, because each one has different definition, we have to work on it. But there is a formation of a network. And the reason that the network is formed is because water under normal condition is far from equilibrium. Okay, and the next thing is that all of us here, otherwise people will not come to the meeting, is that we are convinced that there is a storage and transfer of information. But we don't know how it happened. I mean, we have some speculation. Okay. The idea that we have this is wrong, it's an artist concept. The water do not spin like this, I think. Some people think that this is correct. It's very beautiful for presentation, but I don't think it's correct. And there is a good reason to assume that this is not the case. In any case, I don't want to get into the argument, everybody agrees that there is a structure of order around the nanoparticle and around nanobubble. And this order, the iteration, can induce an order which is up to some distance. Now, the amount of order or the distance of order can be then debated. Emilio has one picture of some structure, nanostructure, that then combine together and the nanobubbles make an anchors for this structure. In any case, I think that what we agree with, or the agreed upon concept, is that if we have this large object, there is exchange interaction that can be mediated via the water or via some additional structures in the water. This exchange interaction, if you take the elements of the physics and mathematics of self-organization and assume that water is far from equilibrium. Why is it far from equilibrium, this glass of water? Because the conditions here of the different field, the acoustic field, the temperature, the electromagnetic, and the temperature in the surrounding is different. So there is all the time imbalance in standing water. This is the driver for the fact that you have this self-organization. Of course, if you have nanoparticles can stabilize this order even more. Okay, so this is another part of it, and this is some experiments that we show that is very puzzling. That treatment, electromagnetic treatment of water triggers effects which are similar to the fact that you have nanoparticles. So the idea is that the water, the electromagnetic radiation really generate this order or the same effect, but if you don't have nanoparticle, it is less stable. The existence of nanoparticle can stabilize the effect and can amplify the effect of the electromagnetic radiation. Just to show you briefly some of the effect, this is, for example, something which is a very nice experiment. You see here, this goes back to Tesla. This is an open coil, so it's the scalar Tesla effect. This is the water, the fluid in which you grow the bacteria. Of course, it has a test tube and not like this, and this is a test tube, just an artist's presentation. You just put it like this. This is just a filter of the external electromagnetic radiation. So you're left with more ordered electromagnetic radiation. You wait 3 hours. You grow the bacteria overnight in a test tube. Then you take the bacteria and inoculate them and look at the differences. I'm very puzzled by the fact that not only there is a difference in the pattern, but if you look at the chirality here in this way, the chirality here is reversed. Which we have some thoughts, but we don't understand. This is another example. This is more dramatic example, because this example is that you add to the fluid antibiotics. Not in the little level, so the bacteria can grow, but you see that when you have the electromagnetic radiation, the bacteria are able to cope with the antibiotic with a much better way. And that is a bit alarming, because we all the time exposed to electromagnetic radiation outside. And I'm not sure that our body can use it as efficiently as, for example, cancer cells or the bad bacteria. It's something to be tested. You can do also gene expression. These are 2 types of treatment. This is control, and you see that there are significant differences. Okay. So now we go to the reflections. So this is the more or less to share with you some of the elements. And now let me talk about some reflections. One reflection, this is what we have. Again, this is an artist concept. It's not correct. They don't tell like this, okay? This is just to draw the attention that there is some order. In principle, and this is also principle is an artist concept, if you assume that you have 10 to the 12 nanoparticle in 1 milliliter, if you take a cup of water, okay, in principle, you can store amount of information which is equivalent to 1,000 brains. In principle. Okay, it's not correct because the brain doesn't store information according to the number of neurons and so on. So this is an artist concept. But in principle, you can store quite a bit of information. However, if you can store, we don't know how, as I reflected how you store it, and the amount of information that you can store that will be really orthogonal one to the other, so you can store more than one piece of information, you have to store it in a more complicated way. So one of the challenges that I'd like to pose to people, like you, is to see if we can store more than one piece of information, or if we want to store another piece of information, it will erase the information before. Why do I mention it? Because if we really want to talk about memory, this is one of the conditions. Part of my research is in neuroscience. And one of the challenges of people in neuroscience is to take neuron in a dish and to try to imprint memory. So people do treatment and they succeed to do changes in the activity. But then when you treat it again, you do another change. It took us many years to find a way how to imprint one pattern of activity and then imprint another pattern of activity without erasing the previous pattern of activity. So that's, I think, a challenge, because unless we do this, we cannot really talk about memory in the sense that you can store more pieces of information and not just, it's still very fascinating, store one thing. So that's a thing, and I think that such experiments will also be critical to get a better idea of really what's going on. This will also help us to know how and if information can be erased. In the brain, for example, you cannot erase information. Succeeded that you can store, you cannot erase information. What I mean, if you cannot erase information without damaging the brain. Of course, you can damage the brain. For those of us that believe that memory is in the brain, you can store information and you cannot erase it by giving some stimulation without specifically erase this piece of information without damaging the brain, which is And it's not clear what's going on here. So, how much we have time?
You have about 7 minutes.
7 minutes. Well, that's a movie from Austrian movie, but you see me in real life, I'm not sure. Anyway, so the effect of cellular phones, one of the things, and the question for you that I didn't have time to ask, did you try cellular phone to see how it affects the... Oh, okay, so I'll show you how it affects the bacteria brain. So, this is a control, conditions that was hard for them to work. You have cellular phone, 5 minutes, and then put the bacteria in the fluid. And it's not, there is no heating of the fluid, okay? It's not a heating effect. And then you grow the bacteria. And that's afterwards, and you still see the memory. Okay, that was one of the experiments that was very hard for me to believe. And luckily, thanks to this meeting, water meeting, I met Umiana. And we had a discussion. And she did the experiment. I will not explain this aquaponics because she gave a lecture explaining it. So I just show you the result. This is control and cellular phone. So this is really a very significant effect. Now, this doesn't mean that cellular phone to the brain is bad for you and it's good for you. But it means that it has an effect. Okay, now comes the experiment, the additional one, that I think is very important. Before I knew about Luke experiments, I thought independently without talking about it, how can it be that you have transfer of information? And I told you that the motivation was the spore of bacteria. So the idea was, can it be that we take, and Umiana mentioned it, but I mentioned it again. So you take a test tube with no antibiotic, put it in another test tube, larger one with antibiotic, and can it be that some of the information will be transferred? I don't know how. I have some speculations, but so this is the experiment, and we also tested it with electromagnetic radiation, with the coil to see if it's amplified. So these are the cases. You see, this is 4 and 6. No antibiotic, but inside the test tube with antibiotic, and here is no antibiotic. These are the results. This is with antibiotic. This is no antibiotic inside a test tube with antibiotic. This is no antibiotic in a test tube with antibiotic with a coil, so you see that the effect is stronger. But what I'm very happy about that if you look closer in the initial dose, you see that there is some differences between this one and this one. So you see here this darker dot that is like the control. You don't see it here because you don't expect, at least I didn't expect that the information will be or the effect will be exactly the same. Maybe I have to naive. But that was fascinating enough. And luckily, this is to show you reproducibility. Okay, and this is to show you this kind of effect. Okay, so Miana told you about this experiment. I don't know if we ever publish it because I still want to be in science, but this is the experiment and this is the results. This is the many repetition, and Omiana explaining that you see that as you go, you don't go all the way to this case, but you go over here. This is with a closed cup, and this is with open cup, and you have the same effect. Say that this is the nice thing about it, I believe, if one can say good words about his own work or our work, that there is a different type of information, and now we have more than one type of experiments, and it's very important to do this. By the way, the experiment that I did last night, we tried to transfer also alcohol, like this, and we did it, and then we opened and really could smell the alcohol, and it turned out that the technician was confused with the test tube. For one coffee, we went immediately coffee, and we were very happy with the outcome. Then we tried it again, it didn't work. Anyway, talking about healing, this is the idea. You put your hand, actually we don't put the two hands, we put one hand, and for 15 minutes, and you do the control, and you see this is the control, and this is if you put your hand. Which is, again, it's not somatic effect. Why it can help the body? If I have time, I'll reflect how it can be that it specifically can affect some places when you do Reiki. Right now, I'm just triggering the growth of bacteria, which is also fascinating. Just to show you a control, when I did it, I decided that this is the experiment. I decided that either I have the properties of a regaler or of a healer, or it's a more general phenomenon. And I asked my technician to do it also. She has more fluid in her body than I, and actually in her case the effect was somewhat stronger, because I think that the effect of electromagnetic radiation of her is stronger. Okay, in Kobe we did it like this, and this is the student that did it, and both she did it and I did it, and in Japan I was the larger body mass. So there were differences also between, so it's so sensitive that we could detect differences. It looked the same, but if you look at the parameter, there were differences. So you can see it here again, and you see that the behavior is very similar to what the cellular form does. Okay, just a reminder. Okay, let's me, how much time I have?
You're out of time.
I'm out of time. Okay, so as a conclusion, and then if someone asks me, I'll tell you why I think it helps the healing in a concrete way. That's also on TV. James Bond was right, and he already did the test long time ago. There is a difference between shaking and stirring.
Thank you, that was wonderful. I don't know if it's relevant, but everyone's talking about testing natural water, tap water, and also, I'm just wondering, getting it from the source, like mineral water, are you going to the well and taking it and testing it? Or is it bottled and sitting on the shelf? Do you think that's relevant? And
Okay.
thank you.
That's excellent question. There are differences. And there are people here who are expert in it. Some of the water lose their, I don't know how to say it, but let me say it in a modest way, the biocompatible properties when they take them right from the spring and they move and they bottle and how they bottle, okay? Especially you have water that bottled in plastic, okay? And the water exposed. They're supposed to be transferred in trucks, they're exposed to the sun, they're exposed to many things, so they change. We try to do it in a way that it's, that there is some idea how they came from. And you can see that things change. But the particles remain. Okay?
I guess I was wondering, is anyone testing it directly from the source? Because clearly it's changing.
Yeah, some of the water that they tested from groundwater came right from the source.
And also, do you find it better, have you ever tested like which, ionizer or different types of systems?
I didn't try ionizer. I assume that it can be very helpful for cancer, for things, or other diseases that I didn't have time to talk about. I believe that if you don't have a problem, you just want to maintain normal life, because we came from this water, natural water, with this amount of pH and so on, it should be better for us under natural condition to have water with pH around 7, 7.5.
Thank you.
Okay, so Fiat, you have, and then you.
Thank you, that was very interesting. I just have a quick question regarding the anomalous chirality that you see. Just wondering if the coil direction has anything to do with the chirality in the bacterial colony.
The what direction?
The coil direction.
No, the coil direction is not the effect.
Oh.
We try both right-handed and left-handed. The chirality of the bacteria has to do with the flagella. The flagella move like this, and when they move like this for some time, and then they then they tumble around, they pick a new direction. The bacteria that show the current pattern are long bacteria, they are liquid crystalline, so only the one at the end can turn, and because of this chirality of the flagella, they go in one end. For some reason, for some conditions, they switch chirality, for example, if you increase the temperature. Why it happened for the case that we did, I don't know. But if I may add one thing, I think that the chirality, basic chirality in nature, I didn't talk about experiments that we do in natural water with optical rotation. And we see broken symmetry between left-handed and right-handed. This is very puzzling, because there should not be, according to the ordinary physics, such broken symmetry. I personally think that it has to do with auto and para water, and we tried it with T2O, and you don't see this broken symmetry. And I think it has to do eventually with the natural broken symmetry of water between left and right. And that's what selected the chirality that we see in biological systems.
You showed that in natural water, most abundant nanoparticle is calcium carbonate. But I have read the paper reporting that analysis result in Swedish tap water, carbon organic nanoparticle and silica nanoparticles. How do you think that calcium carbonate is important for function of water?
Okay, it's a good question. By the way, if you notice, some of the carbonate particles, they had a shell. The shell had silica, small amount of silica. And the silica is very important, you see, for doing some of the attachment of H3 or OH and so on. So, now, it differs from different tap water, the exact structure of the particles. But we didn't see particles that are maybe silica. We didn't see it.
Thank you, Ben.
My question is just related to the first one. Because most water now is treated by reverse osmosis. Should it clean from nanoparticles?
Yeah, that's what I showed the treated purified water. So it's different. They don't have, I have some more figures that I didn't show, they don't have the buffering effect of pH. And I believe that drinking this water is bad for you because it's bad for your bacteria. It puts them under stress. And then they send to your body all kinds of toxins and so on.
I think we need to move on unless there is a pressing question. Okay, thank you very much. Okay, so the last presentation in this session, and then we'll have a bit of discussion afterward for a few minutes. We have some time left. The last presentation is the poster winner. And as you know from last night, if you weren't too drunk, the winner was Istvan Huzar. How do you pronounce your last name? Huzar, okay. And here's our winner, and I feel actually very proud to announce his victory last night, because in fact he's the student of my student, so in fact that makes him my grandson.